But when you look at things like microbial algae, the numbers go almost off the charts. It's not clear whether we can get up to 10,000 gallons an acre, but it's quite possible. The problem, in my view, is most people have been viewing algae as farming, not as production. And so we've been designing and altering unique organisms to go from sunlight and CO2 to fuels. And we've done this by looking at the genetic code and species that secrete things. And we've gotten the algae to actually secrete lipids, and we can determine the size of the lipid that they secrete into the media. So this is an example of secreting a C8 and C10 lipid, which can go into any refinery and be cracked into gasoline used as a chemical feedstock for industry. And so now we're looking at these microorganisms as factories, not something we just grow and then have to harvest. We can grow them continuously. I first got the idea from this when we did the Methanococcus genome, this autotroph that works at incredibly high pressures, 1.5 miles deep in the ocean, high temperatures, 85 degrees centigrade, and goes from CO2 to methane with only a little bit of molecular hydrogen. We've been using all these tools in collaboration with BP, looking at biodiversity deep in the earth. And I think this was one of the most stunning experiments that went beyond any degree of expectation. Over 1 mile deep in the earth, the first samples that we looked at under the microscope had such density of biology, more so than we've seen in any part of the oceans anywhere in the world. And it's just stunning. All these estimates that there might be more biomass deep in the earth than we see in the entire visible world looks like it's probably true. These are high temperature organisms, depending on the depth. The biggest surprise to me was it was loaded with spirochetes. Maybe perhaps because they could swim fast and get in and around these different areas. Our first sample was from 50-million-year-old water, and it was dated with carbon dating. We now have a sample 135-million-year-old water from deep in the earth, and it gives us a real shot at looking at isolated environments in terms of evolution. Some of these organisms look very similar to what we found in other places, including in the Black Sea. So here's a sample from a mine deep in Colorado in the Black Sea where Shewinella and other similar organisms have come from. We have a wide variety of organisms now that grow on the surface of coal. The other breakthrough, instead of 0.1% of things that are in the environment can go into culture, we have new unique tools now using the world's first anaerobic cell sorter, microencapsulation, where we've been able to culture and grow unique isolates, getting 80% of them out of the environment now. We're using our genomic sequencing to characterize the entire environment, getting organisms like this that, I don't know if it's clear in the test tube on the left, those are bubbles of methane coming off the coal just by adding a fairly low density of these microorganisms. They break down the coal into acetate and other chemicals. We have methanogens that rapidly take the acetate and metabolize that into methane. We think we can take coal and different sets of microorganisms and make any chemical substrate, which could then be used in manufacturing or go into methane gas. So I've taken you through a journey from the human genome to the environment, back to humans, and then the future with trying to write the genetic code. And we have examples of things with new vaccines, new chemical bases, new sources of energy, that I think it's going to be a very exciting future. Thank you very much.